Hello students, today we will be studying Histology of the Nervous Tissue Part 2. In the Part 1 video, we have seen the features of the neuron and the neuroglial cells, and we also saw the process of myelination. In this video, we will be seeing the slides which are important in nervous tissue, and that is the slides of the nerve fiber, the peripheral nerve transverse section, and the two ganglia, the spinal ganglion and the sympathetic ganglion. Peripheral nerve. It is a collection of many nerve fibers held together by connective tissue. Nerve fiber could be a motor, that is efferent nerve fiber, wherein it's the axon that carries impulses from the central nervous system to the muscle or gland, or it could be a sensory or afferent nerve fiber, that is a nerve process that carries impulses or information towards the central nervous system. Let us now see the peripheral nerve longitudinal section. This shows long cylindrical nerve fibers covered by myelin sheath, prominent oval nucleus of Schwann cells, and distinct nodes of Ranvier. The peripheral nerve fiber is an axon or a dendrite with its covering, that is the myelin sheath and the neurolemma. Each fiber is myelinated and consists of a central axon or an axis cylinder with axoplasm and neurofibrils contained within the axolemma. Myelin sheath is composed of phospholipids interrupted at intervals along the length of the fiber. It is stained by osmic acid and not by the standard hematoxylin and eosin stains. Thin neurolemma sheet is present outside the myelin sheet. The cells of neurolemma, that is the Schwann cells, are neuroectodermal in origin. At the points of interruption of the myelin sheet, the neurolemma comes into intimate contact with the axon, and such areas are known as nodes of Ranvier. Endoneurium is a thin connective tissue layer of mesodermal origin that supports and surrounds the nerve fiber. The potential space between the neurolemma and the endoneurium contains tissue fluid for nourishment of the nerve fibers. This is a low power view of the peripheral nerve fiber longitudinal section, and what we see here are the nerve fibers. A high power view shows us a clearer picture of the nerve fibers. These are myelinated nerve fibers. The salient features for spotting here are presence of long cylindrical nerve fibers, presence of central axon with myelin sheath, and presence of nodes of Ranvier. We then go on to study the peripheral nerve transverse section. Nerve fiber with Schwann cells and the basal lamina is the basic structural unit of the peripheral nerve. Each nerve fiber is surrounded by a thin layer of connective tissue called endoneurium. Endoneurium consists of loose connective tissue with collagen fibrils, few fibroblasts, and occasional mast cells and macrophages. A group of nerve fibers form a nerve bundle or a fascicle. Each nerve fascicle is surrounded by a layer of specialized connective tissue called perineurium. Perineurium is made up of flattened perineural cells arranged in two or more cell layers and externally covered by basal lamina. Many nerve fascicles are held together by dense irregular connective tissue called epineurium. Epineurium surrounds the entire peripheral nerve, and it contains blood vessels that supply the nerve. The blood vessels, that is the vasa nervorum, are present only in the epineurium and in the perineurium. Endoneurium does not have blood vessels. Nerve fibers receive nutrition by diffusion through endoneurium. This diagram here shows us a transverse section of the peripheral nerve. What we see here are the individual nerve fibers, and they are in turn surrounded by a thin layer of connective tissue, that is the endoneurium, and a number of such nerve fibers are held together to form the nerve fascicle or fasciculus, and that in turn is covered by connective tissue which is known as the perineurium, and a number of such fascicles form the entire peripheral nerve, which in turn is surrounded by a connective tissue layer which is called as the epineurium. This is an H&E stained slide showing us the low power view of the peripheral nerve transverse section, and we see here a number of nerve bundles or nerve fascicles, each of them surrounded by the perineurium. A high power view shows us the individual nerve fibers surrounded by endoneurium, and we can see this particular fasciculus surrounded by perineurium. The salient features here for spotting are presence of nerve fibers arranged in fascicles, presence of perineurium that covers the fascicle, presence of epineurium covering the entire peripheral nerve. We now go on to study the ganglion. Ganglion is a collection of nerve cell bodies outside the central nervous system. The ganglia are of two types: sensory or spinal or dorsal root ganglia and autonomic or sympathetic ganglia. Sensory ganglia. These are present just outside the central nervous system. Neurons of sensory ganglia carry impulses towards the central nervous system. Examples being dorsal root ganglia of spinal nerves, sensory ganglia of cranial nerves 5, 7, 9 and 10. The structure of the sensory ganglia. They are pseudo-unipolar neurons, except in the vestibulocochlear ganglia that have bipolar neurons. Neurons have a peripheral process that carries information from sensory receptor and a central process that passes the received information to the central nervous system. A section of sensory ganglia shows large neurons present in groups, mostly at the periphery of the ganglia. Nuclei of neurons are large, vesicular and centrally placed. Groups of neurons are separated by bundles of myelinated nerve fibers. Satellite cells form a complete capsule around each neuronal body. Satellite cells are flattened or low cuboidal cells. The ganglion is covered by fine connective tissue capsule. This here is a low power view of the sensory ganglia. A high power view of sensory ganglia, which shows us the neuronal bodies, and individually they are surrounded by a complete capsule or a complete ring of satellite cells. Salient features for spotting here are presence of large pseudo-unipolar neurons, presence of neurons with centrally placed nucleus, and presence of a well-described satellite cell forming complete capsule around the nerve cell body. We now go on to see the autonomic or sympathetic ganglia. These supply the smooth muscles and glands. Autonomic nervous system shows two neurons: preganglionic neuron, which lies in the central nervous system, and their axons extend from the CNS up to the autonomic ganglia, and postganglionic neuron lie in the autonomic ganglia, and its axon supplies smooth muscle cells and glands. Examples here: sympathetic ganglia are seen in the sympathetic trunk and in the paravertebral ganglia, that is the celiac, superior mesenteric, etc. Parasympathetic ganglia like the ciliary ganglion, submandibular ganglion, otic ganglion, etc. The structure of the autonomic ganglion. They are smaller in size than sensory ganglia, have multipolar postganglionic neurons scattered all throughout the ganglion. The nucleus is large, pale staining with prominent nucleolus, eccentrically placed. Satellite or capsular cells. Very few satellite cells are seen around the nerve cell body, but they do not form a complete capsule around the neuronal body. These neurons are multipolar. Their processes break the continuity of the capsule, and these neurons have synapses in the ganglia. Hence, the complete capsule of satellite cells is not formed here. Ganglion is surrounded by connective tissue capsule. Postganglionic autonomic neurons synthesize catecholamines. Hence, Mesial granules are seen in their cytoplasm. The nerve fibers here are myelinated and non-myelinated. This is a low power view of the autonomic ganglion, where we see very few neuronal bodies scattered throughout the ganglion. A high power view shows us the neuronal bodies scattered all around the ganglion, and we do not appreciate a well demarcated capsule of satellite cells around the neuronal bodies. The salient features for spotters here would be presence of small multipolar neurons, presence of neurons with eccentrically placed nucleus, presence of poorly defined satellite cells. Let us now see the differences between sensory ganglion and autonomic ganglion. The type of neuron is sensory in a sensory ganglion, and it is postganglionic autonomic or sympathetic in a autonomic ganglion. The neuron type is pseudo-unipolar in sensory ganglion, and it is multipolar in autonomic ganglion. Neuronal body shape, this is large spherical in sensory ganglion, small and irregular in autonomic ganglion. Nucleus in a sensory ganglion is seen as large, vesicular and centrally placed, while in autonomic ganglion it is large, vesicular and eccentrically placed. Satellite cells form a complete ring around the nerve cell body in the sensory ganglion. There are very few satellite cells seen in the autonomic ganglion, and they do not form a complete ring around the nerve cell body. Neuronal groups. In sensory ganglion, neurons are seen in groups or clusters. In autonomic ganglion, neurons are scattered all throughout. The nerve fibers are present in bundles that separate the clusters of neurons in sensory ganglion. Nerve fibers are present between the scattered neurons in autonomic ganglion. The type of nerve fibers are myelinated in sensory ganglion. They are myelinated and unmyelinated in autonomic ganglion. Presence of synapse, they do not have synapse in sensory ganglia, and autonomic ganglion has synapses. Thus, we have seen the four important slides which are there in the nervous tissue, that is the nerve fiber, the peripheral nerve transverse section, and the two ganglia, the spinal ganglion and the autonomic ganglion. For further reference, you can read Sontakki Yogesh, Textbook of Human Histology by CBS Publishers and Distributors Private Limited, New Delhi, 2020, page numbers 121 to 135. Thank you.